Deep learning techniques are very powerful at identifying patterns within complex cell image data, but are often criticized as black box prediction, lacking the ability to provide meaningful explanations for the cell properties that drive the outcome. In this project, we reverse engineered a neural network to identify cellular properties that distinguish aggressive from less aggressive metastatic melanoma using label-free movies of living cells. First, we trained a generative deep network to encode compressed representations of single melanoma cell appearances. The network was trained to minimize the discrepancy between input and reconstructed images, entirely independent of any specific classification task. Next, using the autoencoder learned cell representations, we trained a classifier to discriminate between high and low metastatic efficiency tumors and validated the classifier predictions on new melanoma cell lines in mouse xenografts. The power of deep learning to recognize meaningful patterns that escape the human eye is also its biggest weakness: interpretability. What are the physical properties that discriminate high from low metastatic efficient cells? By studying the variability within the embedding feature data, we identified the key features driving classification and used the network decoder to generate a series of exaggerated in silico cell images that amplified the subtle visual cellular properties that appeared to critically define the metastatic efficiency. And viola! Morphing and transitioning cells from high to low revealed the formation of pseudopodial extensions and altered light scattering as emergent properties of synthetic cell images in the high metastatic efficiency domain. These were systematic and specific cell properties associated with the metastatic prediction in our synthetically morphed cells that were validated with the live cell imaging. This is an example of how the application of AI supports the identification of processes that are essential for the execution of complex integrated cell functions, but are too subtle to be identified by human experts.